For decades, Alzheimer's trials cleared amyloid plaques from the brain, yet failed to produce cognitive recovery. This discrepancy points to a specific mechanical reality. The plaque marks the site of a prior cellular catastrophe. Structural data from Gunnar Gora's and Ralph Nixon's laboratories traces the timeline backward, showing the physical destruction of the neuron happens long before any plaque appears in the tissue. The traditional amyloid cascade assumed amyloid clumped outside the cell membrane, but structural data reveals panthos. Amyloid actually accumulates intracellularly, inside multivesicular bodies. When autolysosomes fail to acidify, the cargo causes the organelle to swell, rupture, and extrude a dense core plaque. Under this mechanism, the extracellular aggregate is effectively the inside of the neuron turned out. Plaques act as the gravestones of neurons that died from an internal autophagy lysosomal failure. To halt the disease progression, intervention must target the internal bioenergetic machinery long before the cell physically bursts. This mechanical reality requires replacing the amyloid cascade with the bioenergetic collapse framework. For years, research programs documented autophagy failures in neurons, metabolic crashes in immune cells, and mitochondrial damage across the brain. These are localized measurements of a single integrated system failure. That system is the cellular quality control network, built on two primary pillars: mitochondrial quality control, or mitophagy, and autophagosomal parents. This failure manifests in different ways, depending entirely on the cell type's downstream options. In post-mitotic neurons, failure to clear intracellular debris leads directly to the physical rupture of panthos. In immune cells, like microglia, the exact same failure triggers a catastrophic drop in metabolic output. Alzheimer's disease manifests as a specific, scalable collapse of mitochondrial and autophagosomal quality control spanning across distinct cell populations. This quality control failure is anchored firmly in the genetics of both familial and sporadic Alzheimer's disease, with both risk profiles attacking the exact same cellular pipeline. This flowchart illustrates the macroautophagy pipeline. It relies on two critical operational steps: the initial formation of the vesicle to seal the cargo, and terminal degradation at the lysosome. BIN1 is the second largest genetic risk locus for sporadic, late-onset Alzheimer's. Research from David Rubenstein's lab demonstrates that BIN1 risk variants attack the formation end of the pipeline. They impair the cellular machinery responsible for autophagosome closure, preventing the membrane from sealing around targeted debris. Presenilin mutations operate at the other extreme, acting as the primary driver of familial early-onset Alzheimer's. These mutations attack the degradation end. They disrupt the assembly of the VATPase complex, preventing the lysosome from acidifying and rendering its hydrolytic enzymes useless. Whether the disruption originates at the formation stage due to sporadic genetics or at the terminal end due to familial mutations, the pipeline fails. Undegraded cargo becomes trapped inside the cell, establishing this specific quality control axis as the core mechanistic substrate of the disease. Once a neuron succumbs to this internal failure and extrudes its debris as a plaque, the burden shifts to the brain's microglial cells. Microglia rely on the TREM2 receptor to detect and respond to this debris. The TREM2 receptor functions as a strict metabolic fitness gate, extending beyond its role in simple immune activation. To clear amyloid, the cell requires massive amounts of energy. TREM2 engagement triggers the PAS3K-AKT-mTOR signaling axis, commanding the cell to reprogram its metabolism to fund intense clearance activities. This graph tracks the data from a 2019 cell metabolism paper by Bike and colleagues, mapping microglial bioenergetics during amyloid exposure. The microglia initiate an early glycolytic spike, ramping up energy production to fund the initial stages of phagocytosis. But the signaling axis fails to sustain this metabolic shift. Without adequate mitochondrial support, both glycolysis and oxidative phosphorylation collapse. The cell enters a hypometabolic state of failed salvage. The microglia remain alive, but they are energetically exhausted, entirely incapable of processing the lipid cargo they internalize. The cellular landscape around Alzheimer's plaques reveals a state of profound bioenergetic failure, triggered by the microglial population's inability to fund the demands of clearance. Following this metabolic collapse, these post-homeostatic microglia fracture into three distinct cellular trajectories: DAM, LDAM, and dystrophic. The specific fate of an individual cell is dictated by its total quality control competence, driven primarily by its residual PINK1 Parkin mitophagy capacity. Microglia that retain intact mitophagy turn over their damaged mitochondria rapidly enough to meet the energetic demands of salvage, successfully entering the protective dam state to clear debris. Microglia with impaired mitophagy lose the ability to perform fatty acid oxidation. Unoxidized lipids accumulate in cytoplasmic droplets, resulting in the lipid gridlocked LDAM state. If mitophagy fails entirely, the cell can no longer maintain its mitochondrial population. This total loss of quality control shatters the cell's morphology, leaving it in a terminal dystrophic state. This progression raises a mechanical question. How does an energetically exhausted, gridlocked cell manage to actively destroy the surrounding neural tissue? As mitochondria capacity fails, damaged mitochondria accumulate. These dysfunctional organelles leak reactive oxygen species and damage-associated molecular patterns straight into the cellular cytosol. Research from Michael Henneke's lab identifies the effector gate that weaponizes this leakage, the NLRP3 inflammasome. The leaked mitochondrial molecules physically dock with it, triggering activation. In response, the complex ejects a barrage of interleukins, ASC specs, and matrix metalloproteinases into the surrounding environment. The destructive neuroinflammatory cascade of Alzheimer's disease stems directly from this mitochondrially gated inflammasome trigger. These cellular breakdowns operate within a wider biological context. Martin Picard's framework of mitochondrial allostatic load scales this mechanism from the individual cell up to the entire organism. Mitochondria act as the cellular integrators of a person's systemic biological state. They continuously process inputs from the endocrine system, metabolic pathways, and psychological stress responses. Chronic systemic stressors like insulin resistance, severe lipid dysregulation, and sustained glucocorticoid elevation place constant strain on the cell. Over decades, this cumulative load degrades the internal quality control network. This systemic connection clarifies the clinical phenomenon of cognitive resilience, where certain patients carry heavy amyloid burdens in their brain tissue but demonstrate zero cognitive decline. These individuals maintained systemic bioenergetic trajectories. Their lifelong metabolic habits minimized allostatic load, preventing their cellular quality control machinery from crossing the threshold of terminal failure. The quality control collapses observed in the brain act as localized manifestations of the organism's lifelong systemic metabolic burden. Targeting downstream endpoints, like clearing extracellular amyloid or applying direct inflammasome inhibitors, will only yield modest results because these approaches leave the upstream substrate failure completely intact. A viable therapeutic response requires Michael Ristow's principle of mitohormesis. Interventions like NAD-positive precursors, urolithin A, and ketogenic therapies operate by applying a controlled level of metabolic stress to the cell. This controlled stress forces an adaptive physiological response. It triggers the upregulation of PGC1-alpha, coordinating the biogenesis of new, healthy mitochondria, while heavily enhancing the mitophagy pathways necessary to clear out the damaged ones. These treatments actively restore the specific organelle-level machinery whose failure drives the pathology. Any successful application of these interventions must be judged by a specific, measurable readout in the brain circuitry, the preservation of the perineuronal net and the parvalbumin-positive interneuron it ensheaths. Clinical success in Alzheimer's disease requires the preservation of the neural circuit, a state achieved by restoring the cell's underlying bioenergetic core.